Assalamualaikum and salam sejahtera. So lately, there's a lot of news saying that our marine environment had been polluted. But how did they came up with this research and how did they determine the pollutants in our marine environments? So basically, there's a lot of instruments that can be used to determine the pollutants in our marine environment. However, in this video, my group members and I will introduce to you 6 instruments that were used to determine the pollutants in our marine environment. The first instrument that are used to determine the pollutant in our marine environment is polarography. Polarography is also called as polarographic analysis or volumetry in analytic chemistry. The basic principle of polarography is the study of solution or of electrode by means of electrolysis with two electrodes, which are polarizable electrode and a non-polarizable electrode. The polarizable electrode is a dropping mercury electrode, DME, while the non-polarizable electrode is the saturated calomel electrode, SME. The DME is a anode while the SME is a cathode in this process. The image shows what we use in polarographic analysis. The first thing is you need to have your analyte, which is your sample. Your analyte need to be mixed with the supporting analyte and then it need to be poured into the polarographic cell. The mercury inside the mercury reservoir will flow into the capillary tube. At the tip of the capillary tube, the mercury will hang there for about 3 to 5 seconds, and during this period, the positively charged ion from your analyte will be attracted to the mercury at the DME. Please take note that the positively charged ion from your supporting analyte will not be attracted to the mercury. Polarography is used widely employed for the analysis of trace metals in the alloys including ultra-pure metals, minerals or metallurgy, environmental analysis, foodstuffs, beverage and body fluids, toxicology and clinical analysis. While in the biological system, it is used to determine vitamins, alkaloids, hormones, terpenoid substances, and so on.
The second instrument that I would like to introduce is liquid chromatography and mass spectrometer, or for short LCMS. The LCMS is a technology that involves the use of high performance liquid chromatography or HPLC and as well as a mass spectrometer. With both the technologies involved, the separation and detection of a sample is very accurate and efficient. This LCMS is used to detect natural pollutants in the rivers and as well as streams. Next is to know how the instrument LCMS operates. Firstly, a mixture or a sample of molecules is separated according to their physical and chemical properties in the LC column. Then the separated molecules are sprayed in the ion source so that it can undergo the process of ionization. Later, this ionized molecule will go through the mass spectrometer to get detected based on their mass spectrum. So finally, we will be able to obtain the result we require. The application of LCMS is that it can be used in the determination of different components present in fertilizers and herbicides. It is also used to detect phenylurea herbicides. With this kind of high resolution mass analyzers, harmful pollutants can be detected easily.
The next instrument that are generally used for determination of marine environmental pollutants is the antibody-based sensor. Researchers at the Virginia Institute of Marine Science have combined biology and technology to create a biosensor that uses antibodies to detect marine pollutants. The biosensor could be used to monitor for oil leaks and other hazards. The following explanation is about how it works. First, mix the sample of concern and the antibodies in solution. The pockets of antibodies will bind to the free contaminant. Then, we will get the reading from the instrument which shows the degree of binding of that contaminant. The fundamental principle of the biosensor is that the antibodies produced act as a reagent in the biosensor. On the antibodies, there are also a fluorescent tag that gives a quantitative measurement of the contaminant. However, the specificity of the instrument comes from the antibody that is binding the contaminants in the solution. It is a unique way to use antibodies as an analytic device. The instrument is very sensitive as it can detect down to parts per million concentration, which is necessary for environmental monitoring. The biosensor could be used to monitor oil spill. The researchers have developed the antibodies to look at polycyclic aromatic hydrocarbons, which are known as PAH, which are more prevalent water-soluble components from oil and creosote. With other antibodies, the biosensor also works well for TNT. TNT is best known as an explosive material used in military shells and even in underwater blasting. It ended up in the ground or groundwater, which causes environmental hazard.
So I will describe the next instrument, which is GCMS. GCMS stands for gas chromatography and mass spectrometry. Instrument method that combine the features of GC and MS to identify the different substance within a test sample. So this instrument must work by these two components, must use together, allow a much finer degree of substance identification than either unit used separately. This is not possible to make an accurate identification of a particular molecule by gas chromatography or mass spectrometry alone. The fundamental of this instrument is the sample solution is injected into the gas chromatography inlet, where it is vaporized and swept into chromatographic column by the carrier gas, usually helium. Okay, the sample flow through the column, and the compound comprising the mixture of the interest are separated by virtue of the relative interaction with the coating of the column, stationary phase, and the carrier gas, mobile phase. The later part of the column pass through a heated transfer line and at the entrance to ion source, where the compound electron from the column convert to ions. Okay, the type of pollutant that can be analyzed is a organic pollutant. This is because GCMS is the one of the most accurate tool for analyzing environmental sample.
Now we talk about the next instrument, the fluorescence microscope. The fundamental principle of this instrument is that the excitation filter selects the wavelengths to excite a particular dye within the specimen. Then, the light of the excitation wavelength is focused on the specimen through the objective lens. The fluorescence emitted by the specimen is focused on the detector by the objective. In this case, the fluorescence dye we use is 9-red NR. It's a useful dye for staining highly hydrophobic microplastic selectively. That means it can detect microplastic in the study of fluorescence microscopy. After we put the NR into the water sample, the dye will bind onto the plastic surface and renders them fluorescent when irradiated with the blue light. The fluorescent emission is detected using fluorescence microscope. Next, the image analysis allows fluorescent particles to be identified and counted. Magnified image can be recorded and tied to cover the whole filter area, allowing particles down to a few micrometers to be detected. These analyses can be conducted with the assist of CMOS fluorescence camera to ensure a high-quality results. But the image shows that how the microplastic particles are detected by using the fluorescence microscope and NR dye. We can observe that the microplastic pollutants are shown clearly in the image, while the other organic debris like the carapace of amphipod are not affected by the NR dye.
VS spectrophotometer. This is what UV VS spectrophotometer looks like. Function of UV VS spectrophotometer is to measure how much a chemical substance absorbs light. This is done by measuring the intensity of light that passes through a sample with respect to the intensity of light through a reference sample or blank. This basic principle is that each compound absorbs or transmits light over a certain range of wavelength. Basically, spectrophotometer is related to the interaction of light with matter. As light is absorbed by matter, the result is an increase in the energy content of the atoms or molecules. When ultraviolet radiation are absorbed, this result is the excitation of the electron from the ground state towards a higher energy state. Prepare two water samples, the one that we want to observe and the one is distilled water. Put both into a cuvette, then place the cuvette to observe the purity. The spectrophotometer works by passing a light beam through a sample to measure the light intensity with wavelength at range 185 to 400 nanometer. As the light passes through the monochromator, it will increase the intensity. The light will go through the sample and certain wavelength could be absorbed. When light reaches the detector, then measurement of absorption is complete.